And so what is important is this is a live blood analysis from someone in my office. They're unvaccinated. So initially, I was still seeing people last year with clean blood. And then come January, everyone who walked in was contaminated. And people are saying, for example, things that, oh, these are just contaminants on the slide, or this isn't real. So I want you to look at this filament. And the way you know that it's in the blood is you can see that white blood cells and red blood cells have been attached, and they're literally dying trying to digest this stuff, and they're unable to. In a live blood analysis, you place the drop of blood from the finger on the microscope slides within 30 seconds, and this is immediately visible. This is not a contaminant on the slide. This is really in the human blood. So this is important because La Quinta Columna, who they are calling this stuff graphene oxide. I don't care what you call it. We've analyzed it chemically, and it is hydrogel polymers. It can contain graphene. But the important thing is, I believe we're all talking about the absolute same thing. So they heated up a drop of the Pfizer content, and this filamental network has developed. And this is highly, highly correlating with Clifford Carnicom's work. This is cross-domain bacteria, or what people call morgellons. This is June of 2014. And he developed this in a culture with an electrical current. And you can see they look similar. So when I started working with Clifford Carnicom, basically the reason why I did was I saw the similarities between this is in my office, what I saw in someone's blood. This was Mike Adams' analysis of the deceased Vax clot, and you can see that the internal structure is exactly of what I've also seen. Here you can see a bluish quantum dot. And then Clifford Carnicom had done microscopy on these structures for decades, and I saw that they all look the same. So I investigated what are the correlations between it. I wanted to mention that Mike Adams was was the first one to investigate the deceased blood clot. And he found self-assembling carbon-rich polymers with metals in it. It wasn't blood, and we have confirmed all of that work. This right here is unvaccinated blood. These are experiments with Clifford Carnicom. So what we did, we took a blood sample of someone who was unvaccinated, who appeared to have clean blood, and used an extremely low-level electrical current of 10 milliamps, which is something that you would barely feel as a current if it touches your skin. And in 2 hours, the blood was completely transformed into a network of filaments and unrecognizable. So electricity can be a growth fertilizer, basically, for the cross-domain bacteria, which are synthetic biology. What that means is they have crossbred all three domains of life, which are bacteria, they're archaea and eukaryotes. So these are fungi as well as amoeba-like structures. And they've programmed them to create polymer plastics and rubber like hydrogel. And this is something that Clifford Carnicom has shown in the identification of morgellons. These synthetic biology fibers do create toxins that are toxic to the human body. They have artificial blood in them, as well as they have significant adverse effect on the electrical conductivity of the blood. So what we have done is we took C19 unvaccinated blood and actually extracted through experimentation, through the application of an electrical current, these cross-domain bacteria are the origin of genesis for these filaments. Then what we did was we did near-infrared spectroscopy analysis, which is something that is basically allowing to chemically assess the functional groups that are involved in this substrate. So you get you measure the light emission and you can correlate that with certain chemical signatures. So we took the isolate from the unvaccinated blood and basically said, okay, what is in here? And this is what we found. This is the chemical analysis. So you see polyamides and vinyl alcohols are aspects of hydrogels made from plastics. The other thing is for aromatic groups, amide protein groups also create hydrogels. So you can see that we also found silicone. Silicone is an important ingredient of nanowires and nanostructures used for brain chip. This is an absolute match with the analysis of the 2011 brain chip that was found in a meningioma and chemically analyzed by Hildegard Stanninger. So we also found many sulfur groups, which indicates that proteins with sulfur bonds are extremely challenging to dissolve.